Yeah, this is a little bit of a tangent to what we've been talking about with molecular breeding values and with trying to use genomic markers and selection and things like that, that gets us to other areas where looking at the genomic information between these breeds can be kind of useful for other applications. So I hope to give you some insight on that, on some of what we've been doing, looking at the relationships of beef breeds and how we can predict breed using data off of the 50K chip. So just to start out with, I'm throwing up a slideshow to make sure everybody has their basic breed knowledge here and can tell me what these animals are. And I think everybody knows this. Yeah. What's that? Well, yeah, okay. Getting worse already. What? That one I'd say has some indicus in it, a Brahmin type. What's that? What are those, right? I think we're getting to where we have no idea. It's not always easy to visually determine. If you were to go out here and go to a feedlot, chances are you'd see a black set of cattle and maybe some red sets of cattle depending on where you go, and you'd have no idea what their composition was whatsoever. Okay, but using genomic tools, we may be able to determine what the breed composition of some of these animals are, and that may be useful in areas that we haven't looked at yet. I'll talk about this just a little bit more later, but in areas we haven't looked at yet with this using marker information for selection, well, there may be tools where if we knew the breed composition of the animals are, we could use that in our management systems, because breeds may be different. They be one of our best indicators of how animals could be different in, say, a feedlot situation. Just as an example, not that I want to pick on one thing or another, but feedlot operators always know to look for a little bit of ear, right? Look for a little bit of indicus in their feedlot situations. If we have knowledge of what the breed makeup of those, that would be one of the best indicators of how those animals should be treated relative to other animals in the feedlot. Okay. I'm going to diverge and talk a little bit about the project that led us to look into this a little bit, the 2000 Bull Project, and you'll be hearing results from this later today a little bit. This just gives you an introduction to something that you may or may not have heard a little bit about already. The 2000 Bull Project was a collaborative effort between researchers here at the U.S. Meow Research Center, involved with the National Beef Cattle Evaluation Consortium, and the breed associations. And basically, the breed associations provided semen for DNA for influential sires within their breed. And here at the center, we ran the Illumina bovine SNP 50, commonly known as the 50K chip, on those 2,000 sires. And the purpose really was for US MARC to provide externally phenotyped animals for use as a training set. Within our own animals as a training set, and then export MBVs into the industry through this 2000 Bull Project as an avenue. The number of bulls from each breed was sampled somewhat proportional to the size of the breed, how many animals there are in terms of herd size of each of those breeds. And breed associations were then responsible for selecting the sires that got used here. And primarily, I'd say that was focused on selecting high accuracy sires from the breeds to basically verify that we are doing a good job in our training, if I go back to the training validation analogy, and influential sires so that if we were able to discover novel traits, we did get a greater gain in exporting those novel trait MBVs to the industry. Well, that's the 2000 Bull Project and what its purpose was, but it has a nice byproduct of giving us a large sample of several of these breeds at the same time and gives us an information about how these breeds vary genomically. This is the overall makeup of the 2000 Bull Project. It does include a few additions. Once in a while, these counts do vary from presentation to presentation. Part of that is because the Hereford is actually increased a little bit here by some samples of bulls from Line 1 Herefords out of Miles City, Montana. The Fort Keogh Research Station, ARS Research Station there. And a few of these breeds are incremented by very small amounts from something called the Bovine Hat Map Project as well. Just so you know that some of these numbers may be a little different than you see in other places. But basically, these representations are even though they're a lot smaller for some of these breeds than for some of the larger breeds, in terms of percentage representation of the genomes within that breed, they're relatively similar and actually favor these smaller breeds more than they favor the Angus, for instance, in terms of percentage of the breed. So if you take that and take those breeds and look at the allele frequency of all 50,000 markers simultaneously, and then look at the correlation between those allele frequencies in terms of genetic distance between each of these breeds, you come up with a pretty interesting diagram that says how are these breeds related to one another, how closely are they related to one another, and how are they different. And I've shown iterations of this before, actually, some in the weight tray project session I think last year, but spend a little more time on it this year, you see some interesting things. If you're not familiar with the codes, AR is actually Red Angus rather than RA. But Angus and Red Angus are understandably the most closely related to each other, the most recently divergent breeds of those 2000 bull project. We have the Continentals here, Charolais, Solaire, Limousine, Bronvie, Simmental, Gilvie, that are kind of in a clump by themselves. Have Hereford way out here, and even further out than the regular Hereford sample, we have those line 1 Herefords I mentioned before, an inbred, more divergent line than the regular Herefords. And then have Shorthorn and Maine, which share some genetics with each other sometimes. We have a Key Angus instead of Chianina. Basically, most of those are 75% average Angus in our sample, so they're closely related to the Angus. Then we Have Brahman way out here, furthest removed with the composites of Brahman with the other breeds in between. Most of it's not too surprising, except maybe the Hereford is a little surprising that it's that far out. Some of that can be explained by what's called ascertainment bias. It's basically that a lot of the SNPs on the 50K marker chip were obtained from a Hereford dominette. But at the same time, if you do take those SNPs out of that, these distances, the Hereford is still more removed from the other breeds, even if you take away those SNPs that were obtained from that dominette sequence. Kind of interesting chart, and gives us a good look at this. But the other thing about this is, given that these breeds are removed from each other, we can probably do a good job of taking a crossbred animal, looking at these 50K SNP markers, and decide what breed these crossbred animals should be. So if we wanted to determine breed composition, there's several potential useful applications. Tara McDaniel just spoke a little bit about using pooling applications. Well, if we were to pool DNA samples from a pen arrival, for instance, coming into a feedlot, we might be able to determine the breed composition of that pen, maybe some other marker-assisted management types of SNPs as well, but for sure the breed composition, and maybe think of how we should be treating those animals in the feedlot if I were a feedlot operator. That would be a fairly low-cost application to do with several animals coming into the feedlot at one time. We could trace individuals to herds of known breed composition. This is an application we've actually done off of this recently. If you had an animal you needed to trace back because of a food safety issue or because of a outbreak issue from some disease, we could narrow down the herd that it came from if we knew that animal had, say, a little bit of chianina in it or something to that effect. Okay. And that could save a lot of money if you've ever been involved in these traceback things before, and maybe prevent a lot of heartache for some of the farms that are involved. And then it can be used in our scientific applications to basically say we want a control pool that's made up of these breeds, and we just want to take the average of those breeds and compare it to the samples that we know are basically cases. So in terms of what Tara talked about, for example, a minute ago, we could compare our open heifers to some average of all the breeds across all breeds by combining these breed frequencies together. Okay. In order to determine breed composition, basically we exploit those breed differences I showed in that diagram before using a relatively simple procedure where a crossbred animal is just fit in a statistical model compared to the average breed frequencies of animals on this 2000 bull project. So basically we're just predicting based on the 50K genotype of an individual animal what breed composition he was based on which frequencies of breeds that animal is the closest to. And our objective then was to evaluate whether we could accurately determine breed composition of crossbreds based on these bovine SNP50 genotypes. And in order to test this, we used this GPE population, which we talk about quite a bit. It's basically a population made of crosses of the 7 most prominent beef cattle breeds crossed to a base cow to produce F1 bulls that are crosses of those two and F1 cows. And then the population that we predicted were these that we call F1 squares, the crosses of these F1 bulls and cows. Okay. And basically what we end up here with is a large group of animals that are either 2-way, 3-way, or 4-way crosses. And we use them as sort of a test case to see if we could predict their breed frequency where they are 1/4 of each of the breeds that were made up of those, okay? Knowing those already. This is kind of our results from using that regression. And basically we calculated 2 statistics. The R² is sort of like that proportion of genetic variance explained we've been talking about lately. It's basically how good a job are we doing at explaining the variance in the pedigree from our prediction of breed composition. And basically we're doing pretty well mostly. We're around 90% for most of the breeds except Angus and Red Angus. I'll get to that in a minute. The regression right here, this just measures how large we're predicting the composition to be of each of these breeds relative to the actual pedigree composition or the pedigree-based composition. And generally you see, because this is lower than 1, we're generally under-predicting the pedigree composition slightly. Not necessarily a bad thing. It probably means it's just being a little conservative with calling these breeds completely. And the accuracy is reasonable then in all breeds except Red Angus and Angus. This is some of why that is. Basically, since this is a statistical procedure, we come up with a value that would be, this is the % Angus that we predict it to be, and then we also have a value that from the pedigree that this is the % Angus it's supposed to be. And if you subtract those 2 values from one from the other, you come up with a term called a residual. How far away were we from the pedigree-based prediction? Okay, that's the residual. Well, the residuals for the amount being called Black Angus, the American Angus, and the amount being called Red Angus were correlated to each other. In other words, we tended to switch out the amount that we were calling Red Angus and replacing it with Black Angus and vice versa. Okay, they were both going up in animals because of the close relationship we saw in that diagram between those 2 breeds. Okay, so there was a strong tendency to interchange those, and those residuals were fairly highly correlated at about 60%. So going forward, basically, we combined Angus and Red Angus into one composite breed. As a result of that, we ended up getting our accuracy then up to something comparable to the other breeds. We were able to predict that pedigree-based breed composition with about the same accuracy as we were for the other 5 breeds. And the regression also something similar to the other breeds. And this is the plot we got for that composite. It's really representative of what we got for Hereford and all of the other cycle 7 breeds. You see, as I said before, this regression is less than 1. We actually have a line, if you were to fit that, would be slightly less than 45 degrees. That's that under prediction I was mentioning. But the important thing I want you to get out of this is, in general, if we're near 0%, near 20 to about 25%, or near 50%, we do a pretty good job of segregating. It doesn't show very well, but most of the mass is concentrated pretty close right here. We do a good job of segregating at those breakpoints of 0, 25, 50, and 75% where we see these things diverging. Okay, and I mentioned the accuracy was around 95%. You might ask, well, why weren't we able to achieve 100%? Well, at least part of that, this doesn't explain everything, and I won't get into the rest because we're not completely sure. But, but at least part of the problem is that, that chromosomal inheritance in a pedigree-based system is not 100%. If you look at a 4-way breed cross where we have 4 generic breeds, the breeds are red, purple, blue, and yellow here. You know, on a single chromosome, because of crossover events from the grandparental breeds down into the grandparental chromosomes down into the actual individuals, we end up with different amounts of each of the breeds in the chromosomes, okay? So right here, basically, if the sire was a red by purple cross, we had a crossover event, and that chromosome was inherited in 2 segments, part from the red and part from the purple. Or, sorry, purple. My blue and purple aren't very different, but this is supposed to be kind of blue. And we had another crossover event right here then in the maternal side where we inherited part of the blue breed and mostly yellow breed in proportions like that, okay? And as a result, if we had a chromosomal estimate, this is the actual inheritance of breeds for that chromosome, we ended up with a breed makeup for that chromosome of these proportions rather than 1/4, 1/4, 1/4, 1/4. So the pedigree would say it's quarter, 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 quarter. The actual chromosomal inheritance is something different, okay? And averaging across the chromosome, you get closer to that quarter, 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 but you never quite reach it. It's going to vary around that. So the pedigree is not exactly right either, is really what I'm getting at. And we may be doing better than the pedigree in some cases with our prediction, maybe not completely. That's the part I wasn't sure about. Anyway, breed compositions are generally predicted at a reasonable interval, R², but not perfectly. And from analysis with reduced sets of markers, increasing the number of markers does not seem to help after a certain point. Basically around 15,000, we really saw a diminishing return. We did do this with the markers that are on the Illumina 3K off of this 50K and ended up with about 83% accuracy. So we see about a 6% drop in accuracy if we use only 3,000 markers. So this application could be done a little cheaper than the 50K and probably do a good job if we saw other uses for this and if people are interested. I know if we were dogs, people would be paying all kinds of money to find out what breed of dog they were. And cattle, maybe that's not quite as true. We need our dog slides back from last year, Allison. So, so anyway, with that, I think this is really interesting. It's another application of our genomic tests. And something else I think we can be looking at besides just trying to improve the value of our selection decisions. Any questions?